Okay, students, we're going to talk about today how to produce a gamete. What we're going to be looking at here is objective 2, compare the end products of meiosis with those of mitosis. Things that we need to accomplish within this little lecture here is we need to summarize the events of meiosis, so going all the way from meiosis 1 to meiosis 2, and then the end products of those things, and then of course explain the crossing over and how it contributes to the production of unique individuals. Okay, so let's go ahead and get started. What we're going to be shooting for today is the basic idea of what is meiosis. So we'll start with meiosis. Remember, we talked about mitosis. Meiosis is simply the process of nuclear division that reduces the number of chromosomes in a new cell to half the number of the original cell. So we're going to take the 46 chromosomes in the original cell, and we're going to create cells that have only 23 inside of them. That's the idea of meiosis. We're trying to cut down the number. And the end result, meiosis will produce what are called sex cells or gametes. Okay, so main purpose: produce gametes with half the number of chromosomes than the parent cell. Plain and simple. It's as easy as that. Right now, gamete formation in individuals. For males, meiosis will result in 4 sperm cells. In females, meiosis will result in only one egg cell, but 3 polar bodies. These 3 polar bodies eventually will be disintegrated and dissolved and repurposed. The one egg cell eventually will be at the end. So we'll talk a little bit more about gamete formation when we talk about recombination, but this is the basic general idea. All right, now, mitosis versus meiosis, this basically answers the objective itself for the most part as far as end products of mitosis versus meiosis. And it's pretty straightforward here. Mitosis will result in only 2 diploid cells, so they have 46 chromosomes in them. They're genetically identical to each other, and they only occur in the somatic body cells. So you're talking about skin cells, you're talking about nerve cells, liver cells, muscle cells. Those are all somatic or body cells. Now, meiosis itself, what we're talking about today, we're actually trying to create 4 cells. And each one of those are haploid or have half the number. So the original parent cell will start with 46. We're trying to reduce it down to 23. Now, what's interesting about meiosis, which is good for evolutionary things, for population adaptation, all those things, each one of these haploid cells end up being genetically different than its host, basically because of 3 different things that we'll talk about. So, but basically they're genetically different than each other. So all 4 cells are not the same. And these are only the sex cells. So we're talking about sperm cells and egg cells. Now, here's a general slide to help you answer and summarize the basic steps of meiosis. So we have 2 main stages, meiosis 1 and meiosis 2. What you'll need to know from this top portion here is that meiosis has 4 stages: prophase, metaphase, anaphase, telophase. Each one of them is symbolized by a Roman numeral 1 because they occur in the first process. Same thing over here with meiosis 2, prophase 2, metaphase 2, anaphase 2, telophase 2, because they occur in the second stages of mitosis. Okay, so meiosis has 2 major steps: meiosis 1, meiosis 2. Now, underneath each of those, I have put together a summary which helps you summarize the basic steps in meiosis. So we're working on the separation of homologous chromosomes in meiosis 1. Again, you have a picture of this in your notes. We're going to try to take 46 chromosomes and reduce it to 23 chromosomes in that process. Now, meiosis 2, we're going to take and separate chromatids. So we have a chromosome just like we did in mitosis, and we're going to split it at the centromere. We take 1 chromatid one way and 1 chromatid the other way. So at the end, we start with 23 chromosomes in this one cell. The 2 resulting cells will have 23 chromatids. So both of these work together. By the time you get from meiosis 1, you have 1 cell that has 46 chromosomes in it. By the time you get to the end of meiosis 2, you have 4 cells that have 23 chromatids in them. Okay, so let's kind of talk about some of the basic things that occur. We'll look at the picture which occurs with it, and we'll go from there. Okay, so here is the basic idea that we're describing and talking about. Again, interphase happens with most cells, But here's meiosis 1, here's meiosis 2. So here's the basic idea. This represents a cell that has 46 chromosomes in it, one cell. As we work our way down meiosis 1, we end up with 2 cells. Notice that we had 4 chromosomes here. We now only have 2 chromosomes per cell. 2 chromosomes. Okay, so we've reduced this from 46 down to 23 essentially. So 4 down to 2 here in this diagram. So we're cutting the number of chromosomes in half. Now, when we get to meiosis 2, we have these 2 cells here move over. These 2 cells right here have half the number of chromosomes. Each of them are called haploid cells. We're going to do a very similar process to metaphase, or sorry, not metaphase, mitosis, and we're going to reduce the number of chromatids in here to half. So what's going to occur is we have 4 chromatids in here essentially, and we're going to reduce that down to 2 chromatids per cell. So we're going to go from 2 cells to 4 cells, and in each one of these cells we only have 2 chromatids. So notice that we have a metaphase here where we're going to separate them out, and anaphase, which moves the chromatids to opposite ends of the cell, and of course we end up with 4 cells here. Each has half the number of chromatids than the original cell, and we should have 23 essentially in each one, 2 in this diagram. Okay, so in your notes you have these same types of ideas going on here. So we need to talk about some simple things here up at the top. Prophase 1, metaphase 1, and anaphase 1 is where we'll concentrate, because that's where a majority of the big action occurs. Understanding meiosis 2 as far as separating it down into chromatids will be the next stages that we'll need to concentrate on. So again, prophase 1, again, going back here, this is the idea that we're going to be setting up. I'll get my laser pointed out here. We're going to be right here in prophase 1. So each chromosome pairs with its corresponding homologous chromosome to form what's called a tetrad. So in this case, what a tetrad is this pairing here. So these pair of homologous chromosomes. So now there are 4 chromatids in this tetrad here. 1, 2, 3, 4. Right? Now, that's kind of interesting because at that point in that tetrad we have a process called crossing over. So this crossing over is when we have genetic information that has been moved, okay, exchanged between chromatids. So this might be the father, this might be the mother. The mother and the father exchange genetic material here, and at the end we have 2 chromosomes that are genetically different from their host or from their parent. Okay. Now, the pairing of homologous chromosomes is key to understanding meiosis. It's the key to understanding crossing over, independent assortment, and of course fertilization at the end. So it's key to understand that the pairing of homologous chromosomes in a tetrad occurs at prophase 1. So in metaphase 1, spindle fibers attach to the chromosomes just like they would in metaphase, and they move the tetrads into the middle of the cell, and it's a random alignment. They can move where they need to. It's just by chance where they get to be put. Okay. Anaphase 1, the fibers pull the homologous chromosomes towards the opposite ends of the cell. So we are reducing from 46 chromosomes down to 23 chromosomes as we get from anaphase 1 down to telophase 1. So independent assortment is what we call that, the separation of homologous chromosomes. A father chromosome can go one way, a mother chromosome can go the other way, or in some other cases they might go the opposite way. So this independent assortment is also a process of genetic variation. So we have crossing over, and we have this genetic variation of independent assortment, wherever the chromosomes are going to line up, and then they separate one way or the other. Okay. Now, here's the idea of independent assortment, possibility 1 and possibility 2. This is what we're talking about. As the tetrad is put together here, all right, these 2 tetrads can be lined up in 2 totally different ways. Right? So you can notice here the second tetrad over here is red-blue. Over here is blue-red. Now, in this case, these 2 different possibilities happen in metaphase. Now, what occurs then in independent assortment what occurs in this independent assortment is obviously either of these chromosomes can line up in any variation that they want. And there's really only two variations. Obviously, it could be blue-red, or it could be red-blue up here. That's an independent assortment. There's no assigned place for it to be. Okay. So as they separate now, you can see in this metaphase 2 here, we have 2 blues and 2 reds. As we get down to our 4 daughter cells, of course, we were reducing to the half the number of chromatids that we were in there before. In possibility 2, like I was saying before, we have 2 different looking combinations here. We have blue-red and then red-blue down here. Notice how the metaphase 2 ends up different. Okay, it was blue-blue, red-red here. Now it's blue-red, red-blue. So this changes the variation of these chromosomes within the 4 daughter cells, which of course causes very different looking cells. Now this of course is only just 2 chromosomes, so these aren't looking very much different, these 2 right here. But in the process of having 23 chromosomes, that's going to be quite different. Okay, so here's the idea of anaphase 1. Again, they just go the opposite ends of the cell, separation of those chromatids. Now, telophase 1 and cytokinesis, obviously we have 2 cells that are formed. All right. Now, prophase 2. Meiosis 1 results in 2 haploid cells. So in this process, we have half the number of chromosomes as the original cell. Chromosomes line up similar to in metaphase, so we have those chromosomes going right down the middle, just like we have pictured here. In anaphase 2, we separate those chromatids and move them to the opposite ends of the cell, just like this process here. And of course, our resulting in the end of meiosis 2 is 4 haploid cells, okay, pictured here. So at the end of all of this, this is where we've gone, prophase 1. We have the tetrads forming here. You can see just barely at the tip of the laser pointer there at the top tip, you can see that we have a little crossing over occurring. Can't really see it very well as we go forward, but you can kind of see it right here. You have this dark purple chromosome has just a small light purple there, and this light purple has a dark purple tip to it there. So we have a little crossing over, and you can see it down here as well. That little crossing over occurs. And again, where they line up is random. Okay, so wherever they line up is a random alignment, and how they move to opposite ends of the cell is called independent assortment. So we end up with 2 chromosomes here, 2 cells here with half the number of chromosomes. As we move into meiosis 2, we take these chromosomes and we're going to separate them out into 4 cells that have half the number of chromatids inside of them. So here we have 46 chromosomes. Here we have 23 chromosomes. Here we start out with 23 chromosomes. Here we end up with 23 chromatids. So that's a pretty good summation of what we're kind of talking about here. Okay, so then we'll talk about genetic recombination later. Thanks.